just want to do a brief introduction for you.
Great, thank you.
Jordan Collin is a field botanist in the CNPS Rare Plant Program who has studied biodiversity conservation and landscape horticulture at Cal Poly San Luis Obispo. While they're working as a student curator at the herbarium on the campus, and also assisting in conserving the federally endangered Gamble's watercress with the Fish and Wildlife Service. Tonight, Jordan will present botanical surveys conducted in the Santa Clara River Utom River Watershed. And we're very pleased to have you this evening. And so share your screen and you're on.
Awesome. Thank you so much, Snowdy. And thank you everyone for coming today. So Snowdy, real quick, it says host disabled participant screen sharing. So something must have happened where I got kicked off of co-host.
Okay, here we go. Gotta find you now. I guess when I left, boy, this is weird. Okay, trying to see you.
The sea of people in here.
Okay. Got it. All right. Did it. Okay. Should work.
Great. All right. Hello, everybody. Thank you so much for being here today. As Snowdy prefaced, my name is Jordan Collins. I'm currently a field botanist in the Rare Plant Program, and I work for the California Native Plant Society. So today I'm going to be talking to you guys about the botanical resources of the Santa Clara watershed. It's going to be kind of on the surface. It's just a huge project, a lot that we're doing. So I'm just going to give you a little taste of what we're up to in this beautiful watershed. So just to start, we're going to orient ourselves to where the watershed is. Where is it in California? So I'm sure we're all pretty familiar with this lovely little map of California. Up here we have the North Coast. Here we have the Sierra Nevadas, the Great Valley. We have the South Coast here, or the Central Coast rather, that bleeds down into the South Coast. And then we have the transverse ranges, and then we have our beautiful desert here. And this little outline here is our watershed that we're going to be looking at today. So just a closer look at that. This is where the watershed is. This is Santa Clarita right here, if you're familiar. Pine Mountain Club is up over here. Ventura is down over here, just to orient yourself a little bit. Here's the Santa Monica Mountains. It's a very interesting place. It's in this kind of crossing of a bunch of different regions of California, so it's a very special and diverse place. And for the purposes of this project, I will be referring to this watershed as the Utom watershed. Utom is a Chumash word meaning phantom, and it refers to the river kind of just evaporating out of nowhere due to its seasonality. It's super cool. There's actually different Chumash peoples that use the word differently. Some Chumash people use it to mean river. Some people use it to mean just any general river. But for the purposes of this project and what the term that our partners agreed upon, we're going to call this watershed Utom. So welcome to Utom. It's a beautiful place. And before we go any further, I just wanted to acknowledge the land that we are surveying on. So I wanted to acknowledge that the land our team has been studying through the lens of this project is the both ancestral and contemporary homes of the Chumash and Tataviam peoples. In the late 1700s, Spanish settlers had arrived in California, as many may know, and they began to construct many missions throughout all of Southern California, thus fragmenting a lot of the territory occupied by both of these communities. The outcome of this was a total decimation of lots of Chumash and Tataviam populations. Many Chumash and Tataviam people perished during this genocide, but many also survived and are definitely prospering as a community today, and we're very grateful that they are here today. So presently, the Tatavian people occupy a very large territory of the Mojave Desert, the Libre Mountains, and northern parts of the San Gabriels. Although they're not a federally recognized tribe, they host a suite of community activities. They provide resources for their tribe members. They also support a tribal government that are actually very active in Los Angeles politics, which is awesome. And presently, the Chumash people occupy an extensive area along the coast of California, from San Luis Obispo County through Ventura County, all the way to Los Angeles County. Several Chumash tribes are actually federally recognized, which is great, and they're a very active tribe today. They do tons of community engagement, and they're very focused on land stewardship, retaining traditional ecological knowledge, and educating the public about their culture, which is great. So there's so many ways that you can support these communities. The first group I want to talk about is the Fernandino Tatavian Band of Mission Indians. They launched this really cool campaign actually called Acknowledge Rent. It's pretty much you can give them money because you live on their land is kind of the bullet point of that. So through this initiative, pretty much any person who lives, works, or visits LA County or is just inspired by the tribe, they can contribute money to the tribe. And what this helps with is it helps fund tribal administration operations, it helps fund their education, language revitalization, culture, wellness programming, and trying to obtain federal recognition. So here I have the website where you can go visit. There's not any set amount if you would like to donate to them. They are a very active community, so that would be a great place to support them. The Sani Inez Band of Chumash Indians are also very active community, and they're one of our partners on this project. This community they're very focused on environmental protection, community outreach, language revitalization, and education. They're really working on this really amazing thing. They're constructing a museum to educate the public about Chumash culture, which is really awesome. I scoped their website like all the way through and I couldn't find a very obvious place where you can like donate money, but there's plenty of ways on their website where you can be involved in different community activities. And their website is here for you, chumash.gov, which is great. Looking forward to that museum too. And the Wish Toyo Foundation is also a Chumash community that occupies the watershed. They are dedicated to cultural preservation, connection to the environment, and educational experiences. If you go to their website, they offered a tiered membership program, so you can give as much as is feasible for you. And this is a great way to also support them. So before we jump into the watershed, I'm going to kind of just set the backdrop for the project. Why are we doing it? Who's involved? What's up with this watershed? So to start with like a couple project details, this really came out of a decades-long legal battle with Newhall Ranch, which many of you may be familiar with. In this image here, this is just an image I grabbed from Google Earth. You can see this very large development owned by Newhall Ranch. And right here is Magic Mountain. Here's I-5, and then this is 126 right here. So the Utom project partners, which I'll talk about our partners in just a minute, they all kind of banded together and sued Newhall, and we received a settlement through this action, which is good and bad, right? A settlement means that they still get to build, which sucks. They definitely have some stipulations and mitigation actions they have to take. But it also means that we got a really good chunk of change to do some awesome conservation work. I think it was somewhere around like $16 million, which is just crazy. So that was a positive outcome of that. And currently the settlement funds are being managed by the Seventh Generation Foundation. Every year the Utom partners get together and they kind of decide how they want to divvy up the next kind of pot of money for the next year or two. And so CNPS, as one of these partners, we were tasked to document the botanical resources of this incredible watershed. So here's a little more about our project partners. We are partnered with the Sani Inez Band of Chumash Indians and the Wish Toyo Conservancy, which I have mentioned, and also the Center for Biological Diversity. And you can go and check out our partners more. We actually have a website for the Utom River Conservation Fund. It's down here. You can go and check that out. And if you go to our website, there's a bunch of tabs at the top where you can just find out who's part of it and different parts of the project. Different partners are working on different aspects of the watershed, like people are working on water quality and wildlife. Yeah, and then one thing here I didn't really highlight, but we actually have a scholarship and fellowship. So you can actually apply for a scholarship if you have a really cool project or an idea that pertains to the watershed. So that's a really great resource for you to go and check out. So now I want to talk about the project timeline and the people. Surveys started being conducted as early as 2017, didn't really hit the ground till 2018 though, and we're still serving till today, which is awesome. And we actually have funding all the way through next year, and we will likely continue to get funding after that, hopefully. So with this task that we were given to document the botanical resources of this watershed, we tried to focus on areas that had very few records, like botanical black holes, pretty much, just to, really get a full scope and a full picture of this watershed. Throughout the timeline of this project, we've had 11 botanists working on it. It's myself, Ellen Dean, Kristen Nelson, David Magny, a couple of my other coworkers. Ben Carter's been very helpful, Annie Zell, and yes. And then we've also had many, many volunteers working on this project. Many of you are here today, and I'm so happy to see your faces. Thank you so much. So these are a couple of my all-star volunteers here. Up here is Dan Horowitz and myself, and we're collecting some bryophytes, which is really cool. I was teaching him how to collect some meaningful moss collections. And then down here, we have my friend Avery Hansen and Betsy Lockhart. We are traversing Fish Canyon, which is north of Castaic Lake. Yeah, and then other volunteers throughout the timeline of this project. I also saw David Torfe is here, so thank you for all your help. Mike Abifara was another person, my friend Leo, and also Jake Bauer has been very helpful with my moss collections and identifications. Here's a couple more friends. This is Jake here, scoping out all the little mossies in this little crevice of this rock. And this is my friend, colleague, and supervisor, Kristen Nelson, and we are going through Pyru Creek looking for some fun plant babies. So before we did any before we went into any field surveys at all, we did pretty extensive literature and database searches. Of course, there are some existing floras in the area. The one that I use a lot is Steve Boyd's Libre's flora, which has been great. We also used Clifton Smith's Santa Barbara flora, Twisselman's Kern flora, and the Moon's SoCal flora. David Magny, pictured here, taking a selfie of us botanizing in Santa Paula Canyon. Also has a lovely drafted manuscript of Ventura County with some great keys in it. So that has also been very helpful when traversing the watershed. In addition to using all the existing floras of the area, we also contacted many local experts and have been in contact with them. Some notable ones that have been personally helpful to me, Rick Burgess, we haven't communicated that much, but your records have been very important to me and my understanding of the watershed. So thank you for all your collecting that you do, Rick. Pam DeVries has been very kind, and she did I think it was a book about the wildflowers of the San Emigdio Mountains, which is kind of the northern part of our range. And she's kind of helped point out a lot of the rare plant species. So that's been awesome. And then Adam Searcy, one of my colleagues, has also been monumentally helpful with plant identification, as well as scouting out localities to look for some cool stuff. So we had our floras, we were contacting experts. We also did a CNDDB search for sensitive taxa in the area. The CNDDB stands for the California Natural Diversity Database, always a mouthful. And that is a natural heritage program that the state of California uses to track all sensitive species in California. So that data has been really helpful for keeping track of our sensitive species. And then we also used CalFlora and CCH2. Many of you are probably familiar with CalFlora. I use it literally daily. If you scroll through my phone on my Safari, it's all just CalFlora. I love that website. And then CCH2 is the Consortium of California Herbaria, and that is where all specimen data is housed, and that's also very helpful. So compiling those two main sources, we have about 39,000 records that we use for our study that have been helpful to inform where our plant populations are. So that's been great. And so what we did with all that awesome data is we downloaded it and we created a geodatabase, which is pretty much just putting points on a map. and kind of using it to geospatially assess plant populations. And we did this through ArcGIS Online, which is an Esri product used for creating maps. So with all that information, we have split up the watershed into subdivisions. So David Magny actually drew all these out. To orient you to this picture here, this big pink polygon here is the actual watershed where water is actually flowing down into the Santa Clara River right here. And all these red lines are delineations of what we're calling bioregions. They're not exactly bioregions. They're more based on prominent geographic features of a given area. So like Alamo Mountain is a prominent mountain right here. So we call this whole area Alamo Mountain. They're also named after like canyons and valleys. Like this is our really large bioregion, and it's the Santa Clara River Valley. So David drew up 54 distinct regions. And this was really helpful because it made everything a little smaller, made them into smaller areas, and it made it just easier to digest the watershed. The watershed is huge, you guys. I'm going to talk a little bit more about the vastness of it soon. But this really helped to orient you to where you were in the watershed and like for giving more specific locality data, which was great. And the idea behind this is we were trying to make miniature floras within the greater watershed to kind of track where all these plants were living. Yes. So our survey methods, this is a very busy map, and it's a lot to look at, but it's all incredibly useful data. This is actually from the end of 2022. I have not updated it with my data from this year. It will have many more points on it. But all these various points are from different people. So these like green triangles, if you can kind of see here, these are all points that CNPS has dropped throughout the watershed. These darker green ones are from the Consortium of Herbaria. So those are actual collection records. And these yellow ones are all from CalFlora records. These pink ones are the CNDDB records. And then, yeah, the NRIS records are these orange ones. So how we did this is we dropped non-random waypoints in natural spaces. And the reason we wanted to drop non-random waypoints is because we're trying to capture as much diversity as we can within the watershed. So essentially what we do is we'd hike a bit. If we saw a plant we haven't seen before, we'd stop and we document it. And then you kind of keep going until you like find another new plant. So that's kind of what I mean by a non-random waypoint. So what happened when we dropped a waypoint is we get our GPS location and we kind of like do a little survey in a 10-meter radius around that given GPS point. And that's just to capture kind of a broad spectrum of plants in the area. And we recorded every plant taxon that we encountered there. And sometimes that was like 40 or 50 plants at one given waypoint. I have definitely done that a few times. So it's really awesome. So let's see. Oh yeah, so things that we also wrote down when we dropped these waypoints is we got really good habitat data. So we got habitat descriptions. We wrote things down about the vegetation communities, maybe something about like the frequency or density of a certain taxon that we're looking at. We also wrote down some great like abiotic features like the substrate that it was growing on, the soil texture that it's growing on, what slope aspect, the physiognomy of the habitat. And then of course, with your general collection methods, you want your general localities so someone could relocate a specific waypoint. And then also like dominant and associate species at that waypoint. So throughout all this time, CNPS has dropped 1,338 waypoints, which is nuts. That's all these green triangles all throughout here. Tons of observations. And within those waypoints, we have observed over 18,000 plants. And that's not species, that's just individual observations of a plant. So like I mentioned earlier, we have like, I think 39,000 records from CalFlora and CCH2. And then you add that with 18,000 observations by us. This is a very robust set of data that is very powerful. On average, about 15 taxa were recorded at each waypoint, which is great. And a lot of the waypoints were merely observational data, but a lot of times we also collected vouchers. So vouchers, of course, were always collected with landowner permission. We mostly have been serving in Forest Service lands, and we have a standing permit with them, which is great. They've been awesome to work with. And when we make these vouchers, we always employ the best CNPS ethics and best practices. And that kind of just entails making sure you have a good enough population size, that you're not threatening a population. You don't want to take literally the last plant. Like, how sad would that be if you extirpated a population because you vouchered it? So anyway, we were very thoughtful with all of our vouchers when we did make them, and we try to prioritize vouchering areas that are very underbotanized and undercollected. And for this study, we collected 2,358 vouchers, and half of them are for me. I collected literally over 1,400 plants. It's insanity, but I love it. They're really powerful tools. Vouchers are extremely important for botany as a science. They are the best piece of evidence that a plant is where you said it was. It is a physical piece of evidence that anyone can go and look at, especially when they're put in a public herbarium. So yeah, this has been a really fun part of my job. And this is an image of a volunteer that was not particularly helpful on my survey, but she kept me great company. This is Ellen Dean's fur baby, Fifi Dean, joining us on a survey. We went to the Mike Foster Bear Ponds. And yeah, she was quote unquote helping me. So after we collect all these vouchers, the end goal is to put it in a public herbarium. And that is so it is accessible to the public, so anyone can go check out these specimens. They can agree with the taxa or not, and they can change the names if they felt so inclined. Also, a lot of new species are described from herbarium specimens, which is really awesome. So we were very fortunate. We were donated 4 lovely herbarium cabinets here from UC Irvine this past year. And we currently have them at the CNPS headquarters in Sacramento. And that is where all my vouchers live in the interim. I have 4 cabinets just full of Utom plants. It is so awesome. It's great to have them all in one place because I can just kind of compare things, really take some time to like really study these organisms, and get better IDs on a lot of them, which is awesome. But like I just mentioned too, this is just the interim. I do not want it to live in my office forever. I want these plants out in the world. I want people to go see my specimens. So ultimately, they will go to a public herbarium, which I have been trickling slowly but surely. And a lot of our specimens for this project have gone to UC Santa Barbara. Many of David's specimens are there, and all of my specimens have gone to Cal Poly in San Luis Obispo. So now I'm going to talk about the watershed and the Utom River. This is the reason for the season. This is an incredible river, you guys. It is 84 miles long, and it is the longest free-flowing river in Southern California. So it's extremely special. And free-flowing just means that there's no dams or anything along the way. So it's an incredibly special phenomenon that this is happening in our neck of the woods. The watershed as a whole is over 2 million acres. That's like so hard to fathom, but it is an insane amount of area to cover, especially with not a ton of people, which is why I need volunteers, which I will talk about later, but I would love more volunteer engagement. I need help. Yeah, I want to say it's like bigger than the state of Delaware, something crazy like that. It's pretty incredible. And it ranges in elevation from all the way to sea level, where, as we just saw on the previous slide, the Santa Clara River spits out to the ocean, all the way to 8,831 feet at the summit of Mount Pinos, which is like alpine, subalpine habitat, which is pretty amazing. This picture here actually is from the summit of Mount Pinos. I tried to get a picture with the ocean to get like the highest and lowest in one pic, but you can't really see it's somewhere over here. You'll just have to trust me on that. And this watershed is just classic California climate, Mediterranean climate with warm, dry summers and cool, wet winters. So if you are unfamiliar with the Jepson ecoregions, the Jepson manual has delineated these lovely little ecoregions that kind of encompass a whole different suite of organisms that live there. So our study area is roughly here. So if we look at a closer up version of that, This map here is the watershed. So this dark blue line is the bounds of the watershed. All these red lines are the bioregions that David delineated. And superimposed on that are these exact Jepson ecoregions. So as you can see, this dark purple one is the Western Transverse Ranges, and that encompasses most of the watershed. That's a pretty large chunk of the watershed. On the northern part of the watershed, it kind of tips into the Tehachapis right here, so it gets a little bit of transition there. Down here, we get some of the south coast, which is awesome. We get some of those coastal species. And then over here on the east side, it kind of dips into the northwestern end of the San Gabriel Mountains. So I really just want to hit it home that this place is incredibly biologically diverse because of this crossing. It's crazy. The Mojave Desert's right here. Then you get transition from like the Sierra Nevadas. You get the South Coast coming down here, and then the San Gabriel's here, and then we get all our coastal species here. So it really is a diverse landscape. A lot of different transitions between different ecoregions happen here, and it's pretty incredible. It also makes it a little tricky sometimes because anything is possible in this watershed. You can totally have a desert plant growing way over here in Ventura County. I have found plenty of those. It's pretty remarkable. So I want to touch on the various substrates that we have in the watershed because plants are very driven by substrates in a lot of instances. We don't have anything crazy like ultramafic like the Klamath ranges do. So we don't have a lot of edaphic endemics, and those are just plant species that are adapted to a specific type of substrate or soil. But we still have some pretty gorgeous geology, I must say. So some common ones that you'll see throughout the watershed is granite with a lot of quartzite in it, as seen here on the left. It's a very acidic rock. The moths absolutely love it, as you can see it there too. On the right, you'll see a conglomerate rock formation. These ones always trip me up at first because it literally looks like a block of cement, but I assure you that is a natural phenomenon. Some other substrates, you got some gneiss rock on the left, but gneiss is a really cool metamorphic rock. I always enjoy seeing that. And then on the right, you have a different metamorphic rock. This is schist from the ridge of Sierra Pelona. Has really interesting layers to it and has a very diverse lichen community, actually. Lots of lichens up there. It's awesome. And sedimentary formations are my hands down absolute favorite. They're always so stunning, and I think by far the most dominant substrate type in the watershed. Tons of different kinds. If you look up here, we have kind of classic sandstone. This is up in the northern end of Apple Canyon. It's kind of in the northeastern part of the range. Here we have some marine sedimentary with all these really interesting little like bubbles in them. So fascinating. And then this right here is pyramid rock. This is just south of Pyramid Lake, which you often pass on I-5 going along the Grapevine. You can actually see here, just next to it is the dam, the Pyramid Lake Dam. So that's a really striking feature in the landscape. And then this is just so otherworldly to me. It's just like, you can literally see the rocks being like pushed up. It's absolutely insane. It's beautiful. And there gets to be like a lot of slate in more areas around this area. So we have a large diversity of sedimentary formations, and many of them are quite epic. So we have great plants and great views in the Utom watershed. Okay, but we are not here for rocks, unfortunately. Well, fortunately. I'm going to start talking about the floristics, which is my favorite part. So first I'm going to kind of talk about like what floristics is, why is it important? I'm definitely going to touch on what floristics is because I've often told people that, oh yeah, I'm a floristician. And they're like, oh, cool, you make like flower bouquets. And I'm like, oh, not really. So it can be a kind of esoteric term, especially if you're not in the botany world. So let's talk about what is floristics. It's pretty much a branch of phytobiogeography. It's the science of identifying the plant diversity in a given geographic area. And it's often describing all the local plant distributions within that given geographic area. And why is floristics so important? It's not a crazy science, you guys. Like, I'm not like a chemist in a lab doing some crazy reactions. I'm not like a geneticist doing some genomics on some like new species. But this is still incredibly, incredibly important. It is the foundational knowledge of what we know about plant species in the wild, and especially their distributions. They're absolutely essential to determine whether a taxon is rare or not to an area. And it is absolutely the baseline information that helps with a lot of land management decisions, helps with a lot of conservation decisions, and things of that nature. So it's incredibly important, and I definitely look at plant distributions and records all the time online. So yes. Great. So now we're going to talk about the vascular flora of this region. This is kind of in like two chunks. So I'm going to talk about the vascular flora first, and then I'm going to talk about the bryophyte flora of the area, which I've just started to study this year, which is awesome. So this is a lovely little picture that I took this year on Mount Pinos. It's just classic Gallium aparini, but with the cutest little flowers and those crazy little hairs. So getting into the vascular flora, we're going to start with some big statistics here. So there are 2,356 taxa reported in the watershed. That is absolutely bonkers, you guys. That is like a little more than a third of all of California's flora. It's really special. There's a lot of really unique evolutionary lineages occurring in this watershed, and every single taxon has its own story. It's a lot of stories to keep track of. They all have a very intricate relationship with the geology, the landscape, with people, with fire, with wildlife. So it's really awesome to be able to track down all these stories and kind of sleuth them out. Throughout the timeline of the project, CNPS has recorded 1,294 taxa. And what's really cool is that since the advent of this project, CNPS has found 36 taxa that were not previously documented in the watershed. So that also goes to show you the importance of studies like this. No one would have known those plants were living there unless someone went out and looked. And sometimes that's all it takes, is just going out and looking. I've been saying this word taxa a lot too, and just to eliminate any obscurity, taxa is plural for the word taxon, and it pretty much just refers to any group of organisms by name. So a family name is a taxon, a species is a taxon. But what I mean here with taxa, we mean terminal taxa, so the lowest possible you can get, so down to the variety or subspecies level. An example of that would be, we have this plant called, it's a popcorn flower, it's called Cryptanthum muricata, and in the watershed we have 3 varieties of it. We have muricata, jonesii, and denticulata, so that counts as 3 taxa for the watershed. So looking at the ratio of native to non-native species, we have a great diversity of native taxa, which is awesome. We have 1,894 native taxa reported, which encompasses about 80% of the flora, and about 462 that are non-native to California, and that's about 20% of our flora. And according to the Jepson manual, they cite that in all of California, roughly 75% of our flora is native and roughly 25% is non-native. So this ratio is slightly higher, but it's not really statistically significant, and it's pretty on par with the rest of California. This is a really cool plant too, by the way. It's Acanthomintha obovata subspecies subcordata. I always flip those, man. Such a cool plant though, so striking with those really interesting spines on those bracts, and is one of our rare babies in the watershed that I got to see with Ellen Dean this year. So looking at some more big picture numbers here, looking at the largest plant families in the watershed, I'm sure it's no surprise, but the Asteraceae, which is the sunflower family, encompasses the most amount of taxa within our watershed. They make up 13.5% of the flora, which is a lot of it. And the largest genus within that is Ericomeria, which is our rubber rabbit bushes and things of that nature. There's 18 taxa of Ericomeria, which is awesome. Got some great scrub diversity. The next largest family is the Poaceae, or the grass family, with 191 taxa, representing 8% of the flora, bromes being very prominent. Then we have the Fabaceae, which is the pea family, and we have 142 taxa, making up 6% of our flora. And lupins are by far the most speciose of the pea family here. Then we have the Polygonaceae, which is the buckwheat family, one of my favorite families, such a cool group of organisms. We have 113 taxa, makes up 5% of the flora, and we have incredible Eriogonum diversity with 59 taxa, which is really awesome. And then our fifth largest family is the Polymonaceae. There's about 98 taxa in the watershed, about 4% of the flora, and the largest genus within that is Gilia. So now looking down at the genus level, so the largest genera, please don't say genuses. I don't know if anyone's ever heard that. It sounds almost inappropriate. The plural for genus is genera. So looking at the largest genera, Eriogonum absolutely takes the cake. It's one of my favorite genera, with 59 taxa reported in the watershed. This top picture here is a voucher that I took of Eriogonum maculatum, possibly my favorite Eriogonum. It's so cool holding them up to the sky like that because you can really get an appreciation for their really intricate and interesting architecture that they can form, especially the annual buckwheats. The next down, the second largest is Phacelia. Then we have the lupins, then gilias, then cryptanthas. And then we start kind of picking up some more graminoids with a lot of taxa. So we get the juncus, carex is down there. We also have tons of astragalus. I think a lot of them's kind of squeaking from the desert province, which is awesome. We have some great onion diversity, and we have some awesome saltbush diversity as well. So I'm going to talk about vegetation communities now. So we actually have a program within the within CNPS. It's the vegetation program, and they go all over California, and they do some great fine scale mapping of vegetation communities. So it's really cool. So we've been doing a lot of like looking at like individual taxa, but they really all come together to be a very beautiful, intricate mosaic interacting with each other in these vegetation communities. So vegetation mapping was a little outside of the scope of this project. So I'm just going to briefly touch on kind of the predominant habitat types that you'll find throughout the watershed. So these are just a couple for you. By far, the three predominant habitat types are shrublands, herblands, and woodlands/forests. Yeah, we have an incredible shrubland diversity. The Libre Mountains is definitely Chaparral City. There's, yeah, amazing shrublands in this area, which you can see here. This is a beautiful south-facing chamizal chaparral slope on the crest of Sierra Pelona Ridge. We also, in our montane areas, we have lots of yellow pine forests. It's kind of like a drier forest on our sky islands like Alamo Mountain, Fraser Mountain, and Mount Pinos. We have extensive riparian woodlands and riparian forests. The landscape here is extremely rugged, and there's tons of really awesome canyons. They're not, a lot of them are not very like wide and gentle. A lot of them are literally fissures in the rock, and they're very tall and steep. So we have great riparian vegetation. And then, of course, in our more lowland areas, we have annual herblands comprising of beautiful wildflower shows. This is on the south face of Libre Mountain with a bunch of poppies in the back here. And then there's some fiddlenecks in the front here. So throughout our time in this project, we've documented 149 vegetation alliances, which is great. And a vegetation alliance is kind of a category created by some of my colleagues at CNPS. It's through the Manual of California Vegetation, which you can view online, and they pretty much ascribe an alliance to a given assemblage of plants based on what species are growing there and kind of their densities. So yeah, 149 vegetation alliances, and there are several more that are actually undescribed alliances. So they're completely new plant associations with each other, which is really awesome. So throughout the watershed, we have 30 woodland and forest alliances. We have 61 shrubland alliances, 58 grassland/herbland alliances. And I didn't include pictures of it here. These are kind of just the kind of dominant things that you can expect to encounter, but we also have a lot of other really special habitats in the watershed. We have vernal pools, which is incredible. There's maybe like 4-ish areas where you can find vernal pools in this watershed with tons of rare plants. Some of them are federally listed. We also have like really interesting canyon seeps and springs. We have beautiful meadows. We have coastal dune systems. We have coastal marshes. There's desert scrubs squeaking in from the Mojave. And then we also have some subalpine habitat on our sky islands, which is pretty incredible. And all these special habitats really add to the diversity of the plant species that we find in the watershed. So now we're going to talk about my favorite things, the rare plants of the watershed. I'm in the rare plant program, so I like to pay special attention to these. This is also kind of why we're doing this project. We were employed to do this to kind of keep track of the sensitive species within the watershed. So if we look at this map here, just to orient you again, this dark blue line is the outline of the watershed, and all these thinner red lines are the bioregion delineations. All these purple points are where rare taxa are existing in the watershed. There's a lot. They're scattered everywhere, and that's pretty typical for California, right? I feel like you just like walk 10 feet and you like stumble upon a rare plant, which is great. And you can kind of see it's really interesting. There's really dense clusters in certain areas, and those are really kind of indicative of the habitat geology and special habitat types that kind of where all these special status plants like to congregate. So that's pretty awesome. So currently we have about 151 special status plant species that are documented in the watershed. 10 of these are federally listed, 9 of them are state listed, and only about 42 of them are tracked by the California Natural Diversity Database, or the CNDDB. And I'm going to talk in a second about why only 42 of them are. It's because the CNDDB has a lot to keep track of, so they really only keep track of the rarest of the rare throughout California. So, tracking rare organisms in California, how is this done? So as I mentioned earlier in this presentation, the California Natural Diversity Database is run by the California Department of Fish and Wildlife. It is a natural heritage program that documents any rare organisms in California, so that includes animals and other organisms as well. It's great. This service is actually not accessible to everybody due to the sensitivity of certain species. As many of us may know, a lot of our charismatic California plants are unfortunately poachable, such as Dudleyias. So this is really sensitive data that needs to kind of be kept under wraps. But it is a service that you can pay for and get access to. We have a really great relationship with CDFW, and we often share data back and forth pretty frequently. So this database is extremely useful for scientific researchers to monitor and track populations of rare taxa. Data included in it usually includes like population estimates, phenology estimates, how many were like flowering or fruiting. It also talks about the general habitat and like how viable that population might be in the future, which is great. And then this is just your reminder, the government is tracking everything. They know when you've paid your taxes, they know if you have paid your car registration, and they know where your rare plants are. So at CNPS, we also track rare plants, which is great. So if you have not heard of this, this is the rare plant inventory, which several of my colleagues on this call work on very diligently to keep it very up to date with a lot of great research that takes them a long time. So this is absolutely a resource you should use. You can access it on rareplants.cnps.org. And just a closer look at that, you can search for any rare taxon in California. And this is just a preview. There's a lot more information kind of down below. But I chose Clinopodium mimmuloides, the monkey flower savory, and there's like 1, maybe 2 occurrences of it in the watershed. Beautiful little plant. Anyway, at the top, you'll see it'll have references to other resources where you can see photos, see collections, distributions, and plant descriptions. Gives you a little bit about the taxonomic information, gives you a rare plant rank, other global and state rankings, and then some other kind of like ecology information here. So I would really encourage you to go check that out. And if you're unfamiliar with how rare plants are ranked in California, they're not kind of just willy-nilly ranked. Like I mentioned earlier, floristics is very informative for this process. We absolutely know, we need to know where these organisms are occurring. So CNPS, we have this kind of like tiered system here. CRPR just stands for California Rare Plant Rank. You'll probably see that acronym a lot more in my slides. And it pretty much goes in declension of like really hurting in bad condition to like it's doing okay, but still pretty rare. So our 1As are presumed extinct or extirpated in California. Our 1Bs are really rare species. They're rare or endangered in California and also everywhere else in the world. 2As are extirpated in California, but they might be somewhere else, maybe on the continental US or elsewhere. 2Bs are rare or endangered in California, but they're often common in other parts of the United States. Rank 3s are kind of weird. They're kind of on like a, I don't know, list. They often need a lot of review. Sometimes their distributions are not very well known or their taxonomic validity needs investigation. And we're actually working on a couple projects reviewing some of these rank 3s in the watershed. One of them is mousetail. Is that little mousetail? Yeah, Myoceras minimus. There's like supposedly a rare subspecies of it. So we've been doing a lot of surveys and collecting materials to look at its genetics to see if that taxon is indeed a good taxon and if it is rare. And then rank 4s, we have a lot of rank 4s. These are uncommon in California. They usually have a pretty limited distribution. It's kind of our version of a watch list. So I mentioned earlier that the CNDDB only tracks 42 of our 151 rare taxa in the watershed. So the CNDDB is only tracking 1s and 2s because those are they have the most dire situations essentially. The CNDDB does track these a little bit, but not to the fine degree that they do for 1s and 2s. And often too, when you see on a plant rank, you'll see a threat rank attached to it after. If you see like 1B.1, that means it is seriously threatened in California. 0.2 moderately threatened in California, and 0.3 is not very threatened in California. And just to go back for a second, these are kind of just quick synopses of these rankings. It's not like, oh, it's just rare. It sounds so subjective. These are actually based on actual data in science. So it's usually like a 1B. I can't remember the numbers off the top of my head, but it has a very few amount of occurrences. So it is based on actual population data, which is great. So now I'm just going to jump into some of the rarest plants that we have in our watershed. And the first one I want to talk about is the Ventura marsh milkvetch. This is Astragalus pycnostachys variety Lanosissimus, which has the most atrocious Latin name ever. So I will call it the Ventura marsh milkvetch. This is ranked a 1B1, so it is extremely imperiled in all of the world. It's federally listed as endangered, and it's also state listed as endangered. Currently in the watershed, we only have one wild population, and there's one established mitigation site in the watershed. The wild population was kind of a weird story. It was there was like some oil dumping, and somebody like transplanted a bunch of sand on it just to cover up the oil, and then this plant just miraculously popped up in this random like oil dump site. Super bizarre, but really cool. It's a fenced off area on private land, so it's not accessible. The mitigation site is awesome though, that is along the coast. And I actually had the privilege of going to see this plant with Ellen Dean. It was such a treat. It's always so fun to find a rare plant. It's exhilarating. And this is a photo that I took of this plant. It is absolutely beautiful. It has really nice soft leaves. This plant has a very complicated story, and we also have a very large project working on this plant. It kind of needs like an elevated water table in order to really persist. And there's not a lot of good habitats left for that in the watershed. And the site where we found these plants, the mitigation site, they're actually being irrigated right now and also heavily weeded to kind of nip out the competition. But yeah, so we are totally doing a bigger project on this plant too. So if you check out the CNPS website under rare plants, you can read some more about the Ventura marsh milkvetch. The next plant I want to talk about is Navarresia fossalis, or Moran's pincushion plant. This is also a federally threatened plant. It's listed with CNPS as a 1B1. There are only 3 occurrences in the watershed, which I know some of my volunteers have scouted out, and I've scouted them out with them, which is awesome. One of the biggest occurrences is at the Cruzen Mesa, which many of you may have heard of. It's like private land, but it's apparently conserved right now and pretty inaccessible. Um, but anyway, it's a really cute little plant. And I threw this picture in here because this is in a very highly recreated area. And this is kind of the outline of the vernal pool that it's growing in. So that's another thing. It's growing in a vernal pool, very special habitat, and there's not a lot of those in the watershed. And as you can see, there's a bike trail running right in the center of the pool, which is crazy. So they're definitely threatened with recreation, and there's also some development pressure in some other areas. This is another extremely rare plant globally. This is a photo that David Magny took, and I feel like he's the last person to have seen this plant in like the recent past. This is Acanthocyphus parishii var abramsii, or Abrams oxytheca. Super bizarre little plant. It's got these really cool glands you can see down there, and these like really spiny involucres. This is listed as a 1B2. And to my understanding and through some database digging, it is the singular extant population that grows in the watershed. It is known from a single wild occurrence, which is just wild. So yeah, and I have personally surveyed some historical sites, and I was unsuccessful in finding this plant. David Magny did successfully seed bank this plant. CNPS is also part of this project called the California Plant Rescue, which is really awesome. So what it is a bunch of other, it's a coalition of other organizations, and we pretty much go around California and we collect seeds and we bank them of our rarest taxa that occur in California. So in case something were to happen in the wild populations, they could maybe have a little hope. So I'm going to talk about a couple other really rare plants, but I just want to quickly plug that the LA Santa Monica Mountains chapter does not have a rare plant chair right now. So I'm going to need somebody to step up and do that because there are tons of rare plants in LA County, especially. A lot of them are under quite a bit of development pressure. So I would highly encourage you, if you're very interested in rare plants, you should totally volunteer for that position. It's pretty low stakes, and Snowdy could tell you more about it. So now I'm going to touch on some of the endemics to the watershed. Yeah, it's pretty remarkable that we have endemics in the watershed. So endemic and endemism in general, it means it's completely restricted to an area. So I have 3 plants I'm going to show you. They're the 3 endemics to the watershed, and they only grow in this watershed in the entire world. They're super special and completely unique to this watershed. And the first one I want to talk about is the Newhall sunflower, which we should totally change the common name for. They don't really deserve that name. Anyway, this is Helianthus inexpectatus. It is ranked 1B1. There, you guys, it grows one location on the entire planet. Like, how crazy is that? It's growing out of this like mucky little spring along the Santa Clara River. It's this awesome little rhizomatous perennial. It's got some really interesting involucres happening. And it's growing right next to some Newhall developments. They are doing some mitigation for this plant and roping it off, but only time will tell if it persists, and I really do hope the best for this plant. To my understanding, this plant also has been seed banked, which is great to know, but this is a really special one. It's kind of our poster child for the watershed. It's a really cool sunflower, very recently described too. This is also a really cool plant. This is Ross's pitcher sage, Lepicinia rossii. This was recently discovered within the last 20 years by Steve Boyd. When he was conducting his surveys in the Libre Mountains, he actually discovered it in Ruby Canyon in the Libre Mountains. This is a 1B2, and there's only 3 occurrences of it in the watershed, and those are its only occurrences. To my understanding that it is to my understanding that all these populations are on Forest Service land, which is great. So there's not really a lot of development pressure for this plant, but it is threatened by recreational activities and maybe some mining in some areas. And these 2 pictures here are from Cure Morse. I just wanted to shout Cure out real quick. Many of you are probably familiar with Cure Morse. He's an amazing human. He just finished his PhD working on bush mallows in California, really helping us understand those. He's been really great to connect with. He takes these beautiful pictures, which I'm appreciative of. So if you would like to support Kieran in any way, I know a lot of us utilize his photos for various things. You can go to his website at curiosity.com, super clever. And on his website, he has a great library of all his pictures. He has his Malacathamnus research. And then he also has a Patreon, so you can support Kier on Patreon. But I just wanted to give him a quick shout out because this was the only picture I had of this plant in its full summer glory. And it wasn't too cute. So thank you very much, Kier. And this has been a really exciting find. This is also an endemic to the watershed. So this is an Eriastrum that is currently undescribed. So I actually took a picture of this, couldn't key it out, and I just threw it on iNaturalist. And then David Gowan actually hit me up. He is an expert on various polymones in California. And he's authored many Eriastrum species. And he told me that this is a new taxon. It's very interesting in its habit. It's got really thick stems and it's pretty unique, and it has like glandular tomentose hairs on it. This is it here in fruit. So this plant up here, I actually found right here on the summit of Warm Springs Mountain. And the only other occurrences are just east of it. And with talking with David Gowan about this plant, he seems to believe that it is restricted to this geologic formation here. So if you can kind of see in the map, there's this little triangle that kind of cuts through the landscape, and that is the Francisquito Formation. So oddly enough, this plant is very limited to this unique geological formation just cutting through the Libre Mountains here. So this has been a really cool find, and I'm stoked to see what he produces out of this, but it's just like another special baby that we have in the watershed. So I've talked about just like general rare plants, like rare all over California. I've talked about endemics to the watershed. I want to talk a little bit about some locally rare plants because these are also extremely important for the ecosystems in which they inhabit here. So the first one I want to talk about is a very weird find that I stumbled across actually in Ventura County along Piru Creek. This is the Inyo bricklebush or Burkelia longifolia variety multiflora. And if you look at this map here, a lot of its observations and collections are out in like the desert province and then the mountains over here. And then there's like a couple observations of it down here and right here. So these are super disjunct, and these are the only places you can find in the watershed. And it's just really fascinating. Like you can make up so many stories about this, about how this plant got here. Like maybe it was like following the axis of the Eastern Sierras and just becomes really scattered down here. It's likely really under-detected too because it's pretty nonchalant in bloom. But that was a very interesting find. Like I mentioned earlier, anything can happen in this watershed. I was not expecting to see that plant because it grows way over here. So it's just really awesome. Anything is possible. Another really special locally rare plant to the watershed and to LA County in general is the shin oak, a variety of the Oregon oak. This is Quercus garryana variety Samota. And what's really cool about this, as you can see, it's very prevalent in the North Coast into the Klamath, and then it kind of goes in like the foothills of the Sierras, and then it stops right here in the Libre Mountains. This is the southern extent of its population, which is really special. Yeah, it's just a stunning oak. It's one of my favorite oaks. There's probably about 3 distinct occurrences of it in the watershed. The northwest end of Libre Mountain, the north flank of Grass Mountain, and the north flank of Sierra Pelona Ridge. One thing that I found this year, which was pretty remarkable in talking with some other experts via iNaturalist, is that these oaks down here are actually hybridizing with Quercus berberdifolia, which is really surprising, but not. White oaks, if you know anything about white oaks, they're extremely promiscuous and have very complex population dynamics. So I found a really interesting patch of hybrid swarm between the two, and they likely have some other species kind of ingrained in their genetics there too. But there's definitely, it's like weird stuff going on in general with the scrub oaks in this area, you guys. I encourage you to get to know the scrub oaks and love them. They're always a treat to find, and they're incredibly important to the ecosystems in which they live. And many of you might have heard of this amazing story that came out just this year. So this was a CNPS event. It was so awesome. So this was discovered by a CNPS member named Teresa Spohr. I think I'm pronouncing her last name right. Apologies if not. I have not met her. But thank you, Teresa, for finding this amazing plant. So what happened is that there was a CNPS hike led by the LA Santa Monica Mountains chapters. Many of you might have been on this hike too, which is awesome. The Channel Islands chapter and the San Gabriel Mountains chapter were going on a hike at Mount Pinos, and some of them split off to various things. And Teresa, she's familiar with plants of the San Gabriels and the San Bernardinos, and she spotted this spotted coral root. And she was like, I know what that is. And I just remember Ellen saying, like, I have all these plant lists of Mount Pinos. People have been botanizing it for hundreds of years, not hundreds, but like over 100 years, and it's not on any plant list. And it's quite remarkable. And then when you look at the map of its distribution in California, this was the first time it's ever been observed at Mount Pinos. So this was an extremely special find. And looking at its distribution map, you would absolutely expect it to just connect right here. But previously that was hidden, that was not there, and it was like all disjunct down here. So now we have a nice little bridge there, which is so awesome. But this was just so miraculous. If you don't know about this little orchid, it's an orchid, which is pretty cool, and it's actually non-photosynthetic, and it actually obtains all of its nutrients from fungi, which is crazy. Um, yeah, and it's just so weird that this plant's never been found. Mount Pinos is a heavily botanized area, so this was truly a special find from this year, and it was a total community effort, which was awesome. Rick Burgess and Ellen Dean went back, and I think her name was Carol. I don't remember. I'm sorry. I think she's on this call too. Carol took some sweet pictures, and then Rick grabbed a voucher of it, which is awesome. These are actually my personal pictures. I was in the area shortly after, and I had to see it for myself. So I got some sweet pictures too. So you can actually see a full little story of this that Ellen Dean wrote on the CNPS website. If you just type in like coral root or spotted coral root, and that should come up for you, and you can get the full details there. So yeah, local rarity in the watershed is really important, and also just very fascinating to see these weird disjunct populations show up. Okay, friends, we're gonna talk about the bryophyte flora. Um, I have been on and off into bryology for maybe over 5 years now. I think I got into it actually in 2017. Paul Wilson, a prominent bryologist in California, actually came to Cal Poly and did a Moss Day with us. That's also where I met Amanda Heinrich, which is another biologist out of Santa Barbara who's on this call right now. Hi, Amanda. So I'm just going to geek out about the bryoflora a little bit. This has been a very challenging project, but a very fun, entertaining project. And yeah, so let's just talk about some of the big statistics of what bryophytes occur in the watershed. So I'm going to highlight this right here real quick, actually. So doing my data mining, there was only 72 taxa known to occur within this watershed prior to the study. And I've only started surveying this year, and me and colleagues and volunteers have found 52 additional taxa, which is amazing. That's another one of those things like floristics. All it takes is just looking, and it's so important to know where these plants are residing. So now we know there's approximately 124 taxa. And I say approximately because if you know anything about bryophytes, they're incredibly hard to identify, and you need specialized tools for them. And I have a significant amount that do not have names on them yet. So hopefully new taxa will be rolling in as I get those identified, which is awesome. We currently know of one actually ranked taxon within the watershed, which is really cool, that we have a rare moss. It's not cool that it's rare, but it's cool that we have it. And so far for this project, myself and Ben Carter have collected 667 vouchers, which is insane. My backlog of identification is quite vast at the moment. So I'm just going to talk about a couple of the sensitive bryos that we have in the region. The first one is the listed species that I mentioned to you guys. This is Tortula californica. It's this adorable little moss. It's in the Potaceae, which is a pretty large family of mosses in California. It's really cute with its little awn. It has a smooth awn, these really like glossy looking leaves. And as you can see from this map right here, it's pretty coastal, ranging from the central coast down to the south coast and down into Baja California. It's currently ranked a 1B2, and that's likely because it is endemic to California and Baja. But I've been chatting with Ben Carter about this plant, and he was telling me that they're actually finding a lot more of it, especially along our coastal areas. I'm sure Amanda's probably come across it as well. So he said that it's currently in the process of being delisted or downranked. And I don't know what he means by that because he's Ben Carter and he's silly sometimes. But I personally think it should maybe be revised to a 4. I think that might be more adequate because it still has a very limited distribution and we should keep an eye on it. But I would agree that maybe it's not as rare as we thought. But either way, it is a really special brio that occurs in our watershed. So the next one I want to talk about, this plant is like one of my favorites. It is so adorable. It is called wide leaf crumia moss, called Crumia latifolia. It's not listed on the CNPS inventory at all, but it is, according to NatureServe, globally vulnerable. And most of its threats are limestone mining. But we don't really have a lot of limestone mining in our watershed, so it's not overly threatened. It's more likely going to be threatened by recreational activities. But it's always an interesting find to find it because it likes really wet places, and this is its typical habitat. Really hard to see. This is all like mucky algae, but this is the moss growing right here under submerged running water. So it's getting a lot of oxygen flow. And this just fascinates me. It's this tiny moss clinging to a rock with a bunch of flowing water on it. Like, how is this reproducing at all? Like, how are its gametes swimming around, finding a nearby gametophyte and like reproducing? And how is it getting around? It's anyway, very interesting. Beautiful, huge leaves that look almost like stained glass. They just have this really lovely texture to them. So that has been a really awesome find in the watershed. It is widespread in California, and not particularly rare, but it is a sensitive species that we have in the watershed. So some challenges to biology. First one, they are teeny tiny. So this picture here that I have as a little smattering of mosses on the very tip of my pocket knife. This little itty bitty one here with this tiny little sporophyte is a moss called Gymnostemum calcareum, and it forms these really tight turfs, and it's just got the smallest little leaves on it. So that is definitely a big challenge. It's hard to see them. It's hard to detect them in the field a lot of the times, and you have to find really tight, interesting micro habitats in order to really get some interesting stuff. They're also just really hard to ID in the field. They are almost impossible, not all of them, but most of them you absolutely cannot identify to species in the field. A lot of them you can very readily identify to the genus in the field. But many practicing and experienced biologists have a good field eye for it. And so I've been going out with Ben Carter a lot, and he just taught me so much about just good field ID tips. So these are kind of the main challenges. And another really main challenge is that they're literally microscopic, you guys. They're crazy. They're gametophyte leaves. They're one cell layer thick. So the image on the right is a cross section of a centrichia, which is a really cool moss that grows everywhere. It's a nice pincushion looking one. And yeah, I got that cross section, and it's literally like under a microscope with a needle and like a razor. And you absolutely need that to identify a centrichia. You have to look at the cell patterns in the mid vein right here. And in addition to them being extremely microscopic and needing to look at them under the microscope, microscopes are not cheap. And this is a huge barrier to entry for a lot of people. This is just a quick screenshot I grabbed from the internet. You can see some microscopes, the compound ones that is, range from like $250 to $1,600. Like, that is insanity. a lot of us can't afford that. So that's definitely like a huge barrier for a lot of people. And my recommendation is that if you're looking to utilize a compound microscope, you should absolutely make friends with your local herbarium. They would love to have you. They would love to support you and let you use their resources, or maybe a university might as well. So yeah, definitely some major hurdles with bryology, let alone like the whole different suite of like terminology that you need to describe all these cell patterns and growth habits that these bryos display. So one really amazing thing that I got to do this year is I got to lead a bryophyte workshop with Ben Carter. This was such an awesome experience. So we pretty much gathered a small group of volunteers. Some of us are here today. Thank you for being here. And we just went to a couple places throughout the watershed to scope out some of our bryophytes that live there. So this was in March of this year. It was led by me. I was mostly doing the admin logistics and people corralling, and Dr. Ben Carter, pictured here, scaling some rocks to look at some good bryos. He handled the bryophytes, which was awesome. So yeah, we had about 6 participants. Most of them were beginners. Everyone had a great time and learned a ton. How that went is we did field surveys during the day. So you'll see on the left here is a couple of volunteers and Ben checking out some bryophytes along Pyro Creek, a tributary of Pyro Creek. And then here you can see our little group picture. We were at our friend Devlin's property in the Sespe Wilderness at this gorgeous waterfall. And yeah, we just had a blast. So after our field surveys throughout the day, we went back and we got some good microscope time. So here on the table, we laid out our haul from the day to kind of just like see all the macroscopic differences between the mosses. And then we were learning how to cross-section mosses under the microscope, and things of that nature. So that was a really cool experience. And I will say that I plan to do another bryophyte workshop coming in the spring. It'll likely be April. If you have any interest at all, I would love to have you. This workshop that happened already, we didn't advertise it because it was kind of a pilot thing happening. But I really want to do it again. It was a ton of fun, and it was just great to connect with people too and get people stoked about moss, because not a lot of people are, understandably so, but they're still an incredibly important part of our flora. They make these really cool like biotic crusts that support tons of microfauna. They do some great work for nutrient cycling and soil stabilization, and they're just weird little aliens. It's really cool seeing them under a dissection scope. It's like literally peering into another universe. So I encourage you to stop and look at a moss and say hi next time you see one. So overall challenges to this study as a whole, time. There's never enough time, right? There's only so many places I can get. There's tons of area to cover. So those are definitely factors that limited some of our study. The whole Western transverse ranges, the transverse ranges in general, has very rugged terrain. So some places are just actually inaccessible without falling off a cliff. So that's a little limiting sometimes, which is why we kind of just sample around. And then taxonomic discrepancies in current taxonomic work kind of muddies things sometimes. I've been having a very difficult time with lupins this past season because they don't play by your species rule, and there's some people currently working on kind of untangling those relationships, which is awesome. Some other questionable records in taxonomy in the watershed: the Calochortus clavatus, the club-haired Mariposa lily. Their varieties are, they're kind of wonky, and a lot of people mis-ID them to the variety. So I think some more work needs to be done on those to really delineate those varieties. Um, there's been some weird mystery apunchas I found in the Libres. They're like low elevation chaparral. I can't key it. I've talked to some experts online. Like no one really knows what it is. Some guy wanted to call it Apuncha mojavensis, which is not a valid taxon. So I think apunchas in general are just very messy. So those have been tricky. The tiny little magenta monkey flowers, like the diplicus that are annuals and like the purpley magenta, also incredibly difficult to key. And I think Naomi Fraga is currently working on the monkey flowers in our area because I've raised a lot of questions to her. Devlin has too. And yeah, they've been very tricky. So, volunteer engagement. How can you help with this project? Accompany us on a survey. I love having people join me on surveys. I'm often out alone, and that is so boring. It's so much better to share the adventure with somebody. So I always love taking volunteers out with me. Betsy has gone out with me several times. She's learned a lot from what I can tell. It's great. She has learned how to like press a plant and how to accurately record data for those plants. So I can also teach you a lot, and I'm sure you all could teach me a lot. I love having more eyes with me because everybody just knows different things about different plants. So that's always really awesome. You can absolutely submit data to us too. If you're just out in the watershed and you see some plants, you can get some good habitat and associate data and send that our way. That is monumentally helpful to us. And then you can also just scout locations for phenology checks, which is really helpful for me because I don't live in the area. So I've actually sent Betsy on a couple trips before where she did a little botanizing on her own. She took a bunch of pictures for me just so I can kind of be like, oh, that Coryzanthe doesn't look ready yet. I'll go to a different place instead. So those are actually extremely valuable to me. And another way you can help, the simplest way, join our INAT project. So me and Adam Searcy, my colleague Adam Searcy, just crafted this iNaturalist project. This is probably a little tardy, but it's here now. As you can see, there's 88,000 observations, which is incredible. There's about 2,000 species recorded and tons of people identifying. So you can totally become a member. We just started it, so I haven't built it out too much yet, but would love to see your observations be put on there. Would love if you identified some of our stuff too. If you know the plants of the area, that would be super helpful. So yeah, I'm really excited about this little project. So go check it out on iNaturalist if you use it. And this is honestly the simplest way that you could help us because it's as easy as literally taking a picture and posting it to a social media website. So really encourage you to upload your observations to iNaturalist. And last year, we crafted a report for the watershed. So you can find this report. David Magny has a website of the flora of Ventura County. If you go to venturaflora.com and you navigate
Jordan,
to
I put a
yes,
link to that on the chat.
excellent. Thank you so much, David. If you navigate to this section called Flora, and then if you go to the bottom of the page, it says Botanical Resources of the Utom Santa Clara River Watershed. You can download a pretty hefty PDF, and I do mean hefty, but it has great stuff in it. We put, there's a species matrix where it's like species and which bioregions they occur in. We have a full species checklist that you can peruse through. And then we have little bios of all the bioregions, just little summaries of each bioregion, as well as just like bigger, broader scope for the watershed kind of information. So yeah, be sure to check that out, especially if you want that checklist. That might be very helpful for you if you're just out hiking in the area. Yeah, and I just want to give some shout outs. I am just super grateful to be a part of this project. I'm super grateful to be a part of CMPS and the broader community. It's been such a treat getting to know a lot of you and like having your help and getting a lot of discourse. So huge thank you to my colleagues Ellen, Kristen, and Adam for just putting up with me and for like the emotional and logistic supports of a lot of this project. Massive, massive thank you to my lovely all-star volunteers, Betsy Lockhart, Avery Hansen, Jake Bauer, and Dan Horowitz. Huge thank you to Ben Carter. He's given me a lot of excellent bryophyte knowledge and has just been a really great friend to have. Huge thank you to Devlin Gandy for showing us some sweet sesame spots. If you haven't met Devlin, his brain is like an encyclopedia. He's really awesome to hang out with, and he just knows a lot of things, and he's a great person. I want to thank Laura at the Ventura Land Trust. I've been working with her recently. She's given me some access to some of her parcels to check out their bryophyte communities, which is awesome. Thanks to like the Los Padres and Angeles National Forest for various permits and permissions. Of course, our Utom Conservation Fund partners, the Wish Toyo Foundation actually hosted our bryophyte workshop this past year. So we couldn't have done that without them. And a special thanks to David Magny. He has spearheaded this project, and a lot of his data has been hugely instrumental. I think he also helped a lot with like obtaining this money too for this project and just managing it and pushing it along. So thank you, David, for this project. And with that, I don't know how we're doing on time, but I would love to take any questions. I would love questions about like, oh, where's a good hiking spot? Or if you have more scientific questions, that's cool too. Or if you have a question about a specific plant or land ownership in the watershed. Anything is great. And my email is right there for you too. I would love if you emailed me. I would love if you would come out with me on surveys. I always appreciate the company and see how you can help in any way. You can also follow me on iNaturalist. It's Jordanii, Jordanii. I love just communicating with people on there, and I'm very active on there. So yeah, thank you guys so much. This was a lot of fun.
Thank you, Jordan.
Yeah.
You want to unshare your screen and we'll see if anybody wants to ask questions. I really didn't get any questions in the chat. There were a lot of good comments.
Great.
If anybody wants
poke
to
through 'em.
ask a question, on my screen, the reactions are at the bottom and then you can raise your hand. We've got Andrea and Terry. Andrea, you want to go first?
Okay. You said something about a grass mountain for the shin oak.
Yeah.
Was that the grass mountain that's in the Santa Ynez Valley?
No, man, that happens a lot too. There's like two fish canyons in LA County, and it always confuses me. There's like one by Duarte, and there's a fish canyon north of Castaic. But the grass mountain I was referring to is kind of like near, it's along the which canyon is there? So many? I think it's along San Francisquito Canyon, kind of near the San Andreas Fault area, kind of on the edge of the desert.
Okay, thanks.
Okay, I know I got mixed up. Was that Andrea? Terry,
Yeah.
do you have a question? Maybe not. Carol Bornstein?
Hi, Carol.
Hi, thank you. It was a great presentation. My question has to do with the POACE,
Mhm.
which, is not surprisingly was very high in terms of the number of taxa. But I'm curious, how many of those were native versus exotic?
Yeah, that's a great question. And I haven't specifically dove into that, but I actually do suspect it's almost 50-50 because we do, especially with the bromes, a lot of those are the invasive bromes, unfortunately.
Right.
That's something I'd have to check my database for, though.
Okay, thank you.
Yeah.
Any other
That's great.
questions?
David just put more natives than non-native grasses. Thank you, David.
Okay. Any other questions in person? Okay, I'm just getting a lot of messages in the chat, but they seem to be just saying thanks.
I will take it.
Great
Yeah, and
presentation.
like I mentioned too, this was kind of just like very surface level, broad overview of the watershed. So if you have any more specific questions, again, feel free to email me and I could do more research and track any answers down for you. So yeah, I welcome all of that. David Magny is also a very good resource as evidenced by his profuse amount of comments in the comments.
And definitely download the report, and then you'll get lots of data about where the plant's occurring, and then you can ask your questions after you look at that.
Andrea, do you still have a question, or is your hand?
A new question.
New question. Okay. Hi, Andrea.
You said you wanted to have people go out with you on field trips.
Yeah.
Where do you post them somewhere so people can know that you're having them, or how would someone find that?
Yeah, that's an excellent question. So kind of what you were mentioning was like leading a field trip hike, which actually Snowdy did approach me and said there would be interest for that. So if I led like a proper like field trip hike, that would absolutely be posted likely to the LA Santa Monica Mountains chapter website or the Channel Islands chapter website. or their newsletters that they put out. However they diffuse their information, you'd probably get it that way. But I also just take people individually out. Sometimes it's kind of easier to just take 1 or 2 people out with me, so it's not just as big of an event. So if you were just curious and wanted to go on a hike with me, you could totally just email me and we would just schedule times to meet up. I'm also working on a volunteer website right now, which I can try and disseminate when I have that running, but it would pretty much have my schedule of all my forays. So then you could kind of go in and like pick which ones you wanted to join me on. And it'll also just have all the other resources for the watershed there. So
Sounds good.
Well,
Thanks.
yes, Jordan and I had talked about trying to do a field trip, but you know, that the ball's kind of in his court to decide when would be a good time. You know, we usually do our field trips when it's not real hot.
yeah, no, that
So,
makes sense.
you know, sometime next winter or early spring. So that's something we'd have to decide. And then where would be, you know, a place that would be accessible. So
Yeah, that's been something I've been thinking about too. And honestly, the most accessible place I can think about is either the beach or Mount Pinos, because it has a beautiful paved road that goes all the way like close to the summit. So that's very accessible for a lot of people. Like I mentioned earlier too, this watershed is extremely rugged, so a lot of it kind of requires a little bit of off-roading. So I will have to get back to you on that. And if we do it in the cooler months, we might have to do more low elevation areas because it does snow quite a bit in our higher elevation areas. But there's actually a really nice place on Templin Highway that is quite beautiful. It's just north of Castaic Lake. That would be decent because people can kind of park along the road there. And it's got a quite lovely diversity of habitats and plants. There's like grasslands, there's coastal sage scrub, beautiful riparian woodlands, things of that nature. So that actually might be a good spot. I've taken Betsy there a couple times actually.
yeah, something like that. Because we didn't, not that probably Mount Pinos would be again still fascinating, but we just did that what I think in June. So
Yes, I know, right? Like you all
I'd
just
hate to repeat it, but that's okay. We repeat things all the time anyway. So yeah, I've got to remember to bug you, I guess.
Yeah, I think actually the Templin might be good too. It's fairly non-strenuous. It's mostly a paved road most of the way, and then kind of turns into just a flat dirt road. So I think it'd be pretty accessible for a lot of people to enjoy. And it's so beautiful there in the spring. Great wildflowers over there. So, but yeah, we'll
Great.
we'll talk. We'll figure it out. Would love to do a hike with everyone.
Other than Acanthoscyphus, are there any other plants you're hoping to come across relocate?
Yeah, there's this one plant. It's not like rare, but it's locally rare to LA County. There is a singular collection of this plant. It's Heraclea maximum. I don't know if you've heard of it. It's called cow parsnip, and there's a singular collection in all of LA County from Atmore Meadows in the Libre Mountains. And I went to it this year to try and find it, and I couldn't find it. I was so mad. So you guys should join me, and we'll try and find it. Might be a goose chase, but
Well, that one question I have had because I really, you know, when we first started talking about this, your presentation and stuff, somehow my little tiny brain geographically just thought, oh, we're just going to be along the river.
oh yeah, right?
So
Yeah, you'd think,
we're
right?
just going to be along the river. And then you show me this map and I'm going, Lord, this
Isn't
is
that
huge.
incredible? It's
So,
incredible. All that water trickles down from those mountains and ends up in the Santa Clara. It's remarkable.
so how do you choose? Was it because of you did your homework with the herbaria and all of the and then you found where things had been found before? You didn't
Yes.
just like throw a dart at the map.
Yeah, there's honestly There's a little bit of that, Snowdy. And that's like kind of the botanical black holes that I mentioned, like that have no data, no collections at all. That's definitely like dart in the map, just like, let's go see what we're gonna see, and maybe we'll find cool stuff or not. But then
Right.
you're right also that some of them are very focused surveys to look for like relocating certain plant populations.
Okay. Yeah, because in the past I've heard that people will say, well, we haven't seen this since, 1887 or whatever.
Yeah,
Let's go looking for it.
yeah,
So
totally.
there was some of that, but then some of it is, here's a black hole. Nobody had anything going on in this particular area. Let's go take a look.
Yep.
Okay.
And both
That's-
are of interest because I want current data too. I don't
And
want
how-
like 200-year-old data, you know?
What's the end of this project? I mean, is it when the money runs out?
Yes and no. So, in the words of David Magny, a flora is never complete. We could be botanizing this area for 20 more years and still find new things all the time, I'm sure. But likely, hopefully, it would be great to actually publish like a publication of this flora, because otherwise, what was the point of all this? We put a lot of effort into this, a lot of yeah. So, but yes, ultimately, when the money runs out. And David might be able to speak to this a little better, but to my understanding, right now we have funding till 2024, but we might get more funding after that. They kind of like give it to us in like chunks over time.
So David I can't remember how the the funding came through the settlement, but it is finite, right? It isn't like forever money.
It's fine.
Ultimately, the partners will receive 16.5 million. We basically get 1.5 million each year until that 16.5 million target is reached. I think we're about halfway there. And the partners annually decide, mostly it's invested in stocks and bonds, managed by the seventh generation, as Jordan mentioned. But the partners decide how those monies are spent. And we're being very conservative. I was a representative for CMPS, but now Nick Jensen is the representative. It is funding, from that settlement that I represent CMPS in negotiating that settlement agreement, which I said I'll never settle, but in the end we did. Partly because we were able to extract a lot out of Newhall Ranch that we wouldn't have otherwise gotten through trying to continue to sue them. We sued them all the way to the State Supreme Court. So, but anyway, the partners have a large number of things that we want to get accomplished. And as Jordan said, one of those things is there's an annual scholarship fund for high school students that want to go to college. They have to be residents of the watershed. And then graduate students who want to do some sort of study that applies to the watershed can get actually significant funding, and that's again awarded on an annual basis. So the partners will probably continue that process for a while. Actually, now is a good time to check the Utom website to see if there's probably scholarships being considered right now, and then each year the partners decide how much they're going to allocate to each of the partners. And it's been an equal amount each time. I think this last time we said 250,000 for a 2-year period. And so then we just divide up, CPS divided up what we're going to, different projects within the watershed to do. And the Bryophyte is one of those. Bryophyte surveys of the watershed is one of those. After we completed the vascular flora surveys. And then there's other things like the rare species we're looking at, locally rare species for LA County and for Ventura County to add to the county's lists. Ventura County actually has a formal list that they established through a ordinance. And LA County generally just accepts what we say are locally rare species. But we we have to demonstrate that they qualify as locally rare by using some sort of criterion.
So we're doing everything in the watershed, essentially.
Okay.
a lot happening.
There was a question in the chat from Kristen
Mm.
and asking about good field guides or reference materials, phone apps for botany newbies in the Santa Clara River area, especially along the river in Ventura.
Yeah, so I don't know of any like really stellar like field guides off the top of my head. I know a couple like actual published floras, but that might not be good for a newbie. I mentioned Pam DeVries earlier. She made a wonderful book of the wildflowers of the San Emigdio Mountains. That's a little more north than the river. There's like a little bit of overlap in the species composition, but that's probably more for the northern montane areas of the watershed. Other than that, phone apps, I absolutely love iNaturalist. It gives you excellent suggestions on plant identities, and it's not always right. You should take them with a grain of salt every time, but there's tons of people actively engaged on there that would love to teach you things, and you can totally ping people and ask people about what do you think this plant is, and so that's been a really invaluable resource for me. So I would definitely suggest iNaturalist for beginners. Absolutely. It's great too. You can actually look on a map on iNaturalist and see what other people have observed around you. So that's also a great way to just see what's living in your neck of the woods, essentially.
Okay,
Yeah, and
thank you.
Cal Flora can use a similar way.
Yeah, a lot of people use, somebody just posted
Oh, thank you, Adam. That's
for the
awesome.
Santa Monica's. You know, I don't have any of these apps, but other people I know, besides using iNaturalist, they use Seek.
Hmm. They're actually the same like parent company. They're like the same thing.
Okay, because I've been amazed at it's pretty accurate. We're out in the boondocks and somebody has this, and occasionally it's weird, like
Yeah.
it's something totally off the wall, like it said it's a banana slug and it's a yellow flower, but that's rare. It's just funny.
Yeah, don't ask it to identify a moss. Never correct.
Oh no, you can't do that.
No, it cannot do it. People try, but it's yeah.
Any other questions, guys, or comments? All right, well, we'll try to keep things going and pray that we can get a field trip organized for next late winter, early spring. And maybe it'll be really rainy and we'll get rained out. That's always fun. Or
Adds to the
snowed out.
adventure. Yeah.
And then with rain, there are more plants.
Yeah.
So thank you very much. Thanks for everybody, your input, and we'll be in touch. Take care. Thanks.
Thank you guys for having me. I really appreciate it. And I'm really looking forward to engaging with a lot of you. So enjoy your evenings.
Yeah, it's going to be a wonderful coming year. Okay, I'm going to end the meeting. Bye.